Recording in progress.
So we were discussing about the cell, and in this section we started discussion on what is the cell, what is the size of the cell, and you cannot just tell the size by saying this, that a typical animal cell is about 1/4 of a millimeter in diameter. This is a complicated definition. Okay, so the scientists, they use another method to define the size of the cell. So according to them, they said that the size of an animal cell is about 25 micrometer in diameter, right? And what is micrometer?
Yeah. Very small, -6.
Sorry, I don't
No,
know why your
-3,
voice
minus.
is so low. It's your volume issue, maybe. Because
Let me try to fix it.
okay, 25 into 10 raised to power deci centimilli micro -6. Okay, -6 meters. This much small is the size of the animal cell. Can you hear me now?
Okay, yeah, now is it good?
Yes, it's better now. We were discussing the size. Okay, so
Yeah.
normally the size of the whenever we talk about the length of anything, we say the length is measured in the millimeter or meter. Okay.
Yeah.
So, for example, now if you need to see any object, any like the animal cell, I told you animal cell is too much
Yeah.
small. Like it is the size 25 into 10 raised to power -6 meter. So you cannot
-6.
you cannot see this cell with the naked eye. Okay, you need some aided
Yes.
tools. Some aided tools like you can use the microscope. Okay, and there are different microscopes. The electron microscope. Initially, in the previous times, in the old times, they were using the simple microscope. Okay, then then there was upgradation, and then scientists started using the electron microscope. After that, after further advancement, there was introduction of a scanning electron microscope and tanning electron microscope. So this is electron microscope. There you have.
Yeah.
And then after advancement, there was scanning electron microscope,
Yeah.
then tanning electron microscope, TEM, SEM and TEM. This was simple electron microscope, EM, SEM, TEM. So these advanced microscopic tools, you can use this. Okay, by this you can even check what is inside the tissues. Okay, what is inside the tissue within the cell, what are the things and what are the agents. You can also check all those things by using these microscopes. Okay, what is magnification? We discussed that sometimes it is not easy for us to see anything with the naked eye. It is too small. Our eyes cannot perceive that thing. Okay, so to see that thing, what we need to magnify it. We need to zoom in to that thing. So how can we zoom that image, or how can we magnify that? There is a simple formula for that. Firstly, you must know the actual length. That what is the actual length of this? Like for example, this dot. What is the actual length of this? And what is the measured length? What you have measured? Okay. When you have these two parameters, you can easily calculate the magnification of this. Are you getting this? Firstly,
Yeah.
you need
Yes.
the actual length and also the length which you have measured. You need these two parameters.
And the measure length.
Yes, you need these two parameters to calculate the magnification. And sometimes you don't know the actual length. Okay, sometimes you don't know the actual length.
Yeah.
Then what you can do? Which formula you can use?
Yeah.
If you don't know the actual length, then what you can do?
If you don't know the what's it called, actual length, you have to use measure length over Measure length over magnification.
Yes, measure length over magnification. Then obviously if you don't know this, then obviously you will know measure length and the magnification. You will add these two values and you can calculate the actual length. For example, they have mentioned this is a seed, okay, and they are saying that it is,
Yeah.
they have magnified it by 50 times, okay. It means this statement, it tells that they have magnified it by 50 times. Actually it was like this, the size was like this, and now they have magnified it to 50 times. Are you getting this? Mag into x50. This mag
Yes.
into x50 means they have magnified the poppy seed by 50 times, okay.
Yeah.
Now you know, now you want to
Yes.
know the actual length of this. How you will know? Firstly you will measure it, okay, you will measure it. So maybe you have measured and it was 5, okay, it was 5 millimeter. And what is the magnified? You have magnified it by 50 times.
Yeah.
So you will add these values and you can easily tell the actual length, that is 0.1 millimeter. Are you getting this? But there is
Yes. Yeah.
again, how you will get this measure length? Firstly you have this poppy seed, okay. First step is you place this seed under microscope, you will zoom in, okay, you will magnify it, okay. After that,
Yeah.
once you have magnified it, now you will calculate this length, okay. When you have calculated this length, this is known as measured length, okay. And in this case, what is the measured length? Can you tell me from the formula what is the measured length over here?
The measure length is 5.
Yes, 5 millimeter. And how much
Yeah,
they have
from-
magnified this? The actual was
by
like this.
50.
They have magnified by 50, so that's why 5 divided by 50. So you will get 0.1 millimeter. It means the actual size is 0.1 millimeter. Are you clear with this?
Yeah. Yes.
First step is to zoom in. Second step is what the picture you have zoomed in, you will measure this length, okay, the length of this zoom in picture. Clear? Then you can put
Yes.
in the actual formula and you can find the magnification. Okay, one most important thing, that all the things have same units, okay. Like if you are measuring the
Yeah.
length in the millimeter, you must have this in the millimeter, you have this in the millimeter. The magnification is 50. Actually magnification does not have unit, but this should be also in millimeter, okay. Whatever the length, if you are calculating in centimeter, then actual length should be in centimeter and measured length should also be in the centimeter. Clear?
Centimeter.
Yeah, this is the important thing, that the unit should be same. And to help remember the formula, how you can remember magnification, you see MA, in magnification there is MA, right? MA.
Yes.
So first M is for measured length and second A is for actual length. This is easy way to memorize the
Yes.
formula, okay. Are you clear? Magnification, it starts from
Yeah.
MA. First M is in the numerator, that is measured length, then A is actual length. You can easily memorize the formula by this. Clear?
Yes.
Okay. You have any question, you can ask by till this. If you
Yeah.
Are you clear with this?
Yes.
Okay. Okay, now we are going to discuss about some of the important things which are present in the animal and the plant cell. Now we will compare which thing is present in the animal cell and which is not present in the animal cell, whether it is present in the plant cell. Okay, now first thing, okay, we have discussed what are the common things, what are the common features in the plant and animal cell. Can you tell me? We have discussed
Plant and
The
like
first lecture.
features in plant.
what are the common features.
Plant and animal cell.
Read from here and tell.
Common features in like plant.
Both plant and animal cell have. What are the common features?
It's
Read from
like
here.
have a cell surface membrane. Cytoplasm has a surface membrane. Has cytoplasm. Has a nucleus.
Yeah, they both have cell
Has a
membrane
nucleus as well.
and they both have cytoplasm and nucleus. These three things are common in both plant and animal cell.
Nucleus.
Whatever the type of animal cell, whatever the
Animals.
variety of the plant cell, both will have these three things, okay, present in them. And what are the things which are present only in plant cell? What are the additional things?
What's it called? The plant cell wall is made out of cellulose and has a vacuole and uh, chloroplast to make food.
Yes. Okay, now these were the three common things: the cytoplasm, the cell membrane, and the nucleus. The other things which we are going to see are in the animal cell. If you see this, the animal cell features often relate to heterotrophic nutrition and high rate of metabolic activity. Okay, we can I will tell you what they have mentioned. This is the cytoplasm, okay, the brown color. This is the cytoplasm. Cytoplasm of animal cell is often denser, okay. Denser means it is thick, okay. Denser means
More. Yes.
yeah, it is thick
More.
with many organelles. Why it is thick? Because there are many organelles present in it, okay. That's why it is thick. And there are many dissolved substances also in the cytoplasm. That's why the cytoplasm of the animal cell is thick, it is dense. Clear?
Yes.
Then there are some vacuoles like this. These are the vacuole. There are some vacuoles in it like this. These are secretory vesicles, and over here there is a vacuole. This one is a vacuole, okay. This vacuole, there are vacuoles like this. These are small, these are very small as compared to the plant cell. We will see in the plant cell they can be involved with digestion or with excretion. Contractile vacuole, okay.
Yeah.
Most important thing they have mentioned is that sometimes bacteria enter in our body, or sometimes any infectious particle enters in our body and they try to enter our cells, okay. So when they try to enter our cells, within our cells, inside our cells there are vacuoles present, okay. These vacuoles eat them, okay. They eat those bacteria or
Yeah.
infectious particles, okay. They eat
Yes.
those infectious particles, and this process of eating is known as phagocytosis, okay. What is this known as?
Phagocytosis.
Phagocytosis, okay.
Phagocytosis.
What I am saying is that sometimes infectious particles invade our body. They try to invade the cell, they try to enter within the cell, and they try to have their own you can say the they try to control your cell, okay. So to avoid this, our cell have different things within it. Specialized organelles are there. One of those is vacuole, okay. This vacuole is small and it is present for a temporary time, okay. It is not present permanently. What will happen? This vacuole, it will eat that infectious particle, okay, and after that it will be removed, okay. It will be removed from the cell. Otherwise, what will happen?
Yes.
Otherwise, if it will be burst within the cell, what will happen? The infectious particle, all the infection will be released in the cell.
Will spread.
Yeah, it will spread within the cell. That's why it will be removed. That's why they have mentioned that vacuole is present for the temporary time, okay.
Yes.
Okay, there is one more type of the vacuole that is also used to remove excess water, okay. That is known as contractile vacuole. Simply, we have discussed that vacuoles can do two functions. First is phagocytosis, right, and the other is to remove excess water, okay. That is known as pinocytosis. Let me write it. That is known as
Cytosis.
Phagocytosis means to remove the infectious or other digested food, infectious particles, or the solid things, okay, to remove the solid. any solid that is not necessary for the cell. And pinocytosis mean the vacuole help to remove excess water. Okay,
Excess water.
excess water or any other type of liquid that is not necessary for the cell. Clear? Are you clear with this?
Yes.
Okay, now the next is glycogen. This they have mentioned over here, the glycogen with this color. Glycogen is the storage form of carbohydrate. You know, I told you that sometimes there is extra glucose, sometimes there is the glucose is in excess, and
Yeah.
you don't need that glucose at that moment. So what happens is that the extra glucose converts into glycogen, okay, and this conversion happens within the liver. Okay, whatever food we take, like let's suppose you have taken bread, okay, in the breakfast, and your body
Yes.
needs half of the glucose from that bread. So what will happen with half of the glucose? Then the other half, will that be wasted?
Oh yeah.
That will not be wasted. That will be stored.
Yeah, it will not be.
That will be stored for the future purpose. Okay, just like we have if you have money and you go to the market, half of that currency is used and half of the money is left behind, so you will waste that money.
Yes.
You will
No,
not
no.
waste it, yeah, right? What will you
No.
do?
Yeah. Keep it for future.
Yeah, you will keep it for future. Just like that, glucose is the energy currency of our body. Okay, it gives us instant energy. So when you are taking the carbohydrates, what is happening is basically that glucose, it is that carbohydrate is converting into glucose, then you are utilizing that glucose, and the extra glucose is converting into glycogen. Okay, so we say that glycogen is the stored form of glucose. Okay, it is the stored glucose form. Clear?
Yeah. Yes.
Okay, then you have secretory vesicles. For example, they have mentioned this one. These are secretory vesicles. Okay, what are their function? They contain cell products such as hormones and enzymes that are much common in animal cell. Okay, in these vesicles, the hormones cannot directly release in the cell. They are present within these vesicles. So these vesicles have hormones, okay, as well as they have enzymes. Clear?
Yes.
Okay, then you have the other thing is animal cell. Okay, these are the four things which we have discussed. Now they have mentioned animal cell features often relate to heterotrophic nutrition and high rate of
Heterotrophic.
metabolic activity. What is heterotrophic mode of nutrition? You know about this?
Yeah, you told me that.
Hmm?
Heterotrophic is like something
Hmm.
that they eat like what's it called, like consume like by plants, you know.
No. Heterotrophic means heterotrophic.
Like cannot produce by its own food, like
Those are
it's not like
heterotrophs.
plants.
Yeah, those are heterotrophs. You are right. And heterotrophic nutrition means they rely on variety of food. Okay, they don't rely on just
Yeah.
one kind of food. Heterotrophic means they rely on different kind of food. Okay,
Different types of food.
different
Types of food.
Yes. And the other thing is, as I told you, heterotrophs are those which cannot prepare their own food. Okay, they rely
Yeah.
on others for food. Clear?
Yeah.
They rely
Yes.
on others for food. And now they have mentioned there is high rate of metabolic activity within the liver, just like the liver cell. You know what is the metabolism process? Simply, what
Metabolism.
is metabolism? Yeah.
Like the process of metabolism is I'm not sure, but like metabolism means like the chemical reaction
Yes,
that
exactly. That are happening
in your
within the,
body,
yeah, within the cell.
yeah, to break down like proteins or
Yes,
fats.
exactly. Let's like this glucose. Your cell cannot utilize glucose as it is. It needs to convert
Yeah,
this glucose into ATP, right?
yeah,
Similarly, there are proteins. Your cell cannot rely on proteins. It need to convert that protein into amino acids, right? And then from those
yeah.
amino acids, your cell use those amino acids. So this is known as metabolism. Okay, metabolism means the chemical reactions which are happening in your body. That is metabolism. Clear?
Yes, yeah.
Chemical reactions that are happening in your body, in your cell. Basically, we can say in the cell. Okay. Then these are some of the organelles which we have discussed. Some of the things, not organelles, some of the things which are present in the cell. You can say organelles, but they have different functions. Okay, then the absence of the cellulose wall mean that animal cell may be very irregular in shape. Yeah, this is very important point. You know that we have discussed that in animal cell there is absence of what? Cell wall. Okay, absence of cell wall. In animal cell there
Cell
is
wall, yeah.
no cell wall present. It means
Yeah.
the shape will be what? Shape will be irregular. Okay, the shape
Irregular.
will be irregular. Cell wall gives the regular shape. So it means the animal cell have a irregular shape. Okay, if I ask you in the
Mm.
test what is the reason behind irregular shape of the animal cell, then what will be your answer?
Wait, what's the question?
Hmm?
What's the question?
I am saying, what is the reason behind the irregular shape of the animal cell?
Because like the amount of what's it called? Like the
Yes,
wall
cell wall. Yes,
cellulose.
simply
Yeah, cellulose.
there is no cell wall. Okay, there is no cell wall in
Yeah.
it. That's why it has an irregular shape. The amount of cytoplasm that can be controlled by the nucleus is limited. So that animal cell may be quite small, about 25 micrometer in diameter. This is very important. They are saying the amount of cytoplasm that can be controlled by the nucleus is limited. Nucleus actually, you know, the nucleus is controlling your cell, right? The nucleus is controlling your cell activities. This nucleus, this is the nucleus.
Yes.
They are saying whatever the content present in the cytoplasm is controlled by the nucleus. Okay, whatever the content, whatever the things present in the cytoplasm, whatever the amount of that liquid in the cytoplasm, that all is controlled by the nucleus. Okay, again read from here. They have mentioned the amount of cytoplasm that can be controlled by the nucleus is limited. Okay, why it is limited? Because you know the cell can burst because there is no cell wall. Are you getting my point? There is no
Yeah.
cell wall. So if there is more content within the cytoplasm, what will happen? The cell will burst. Okay, so to avoid this, nucleus only allow limited things. Okay,
Yeah.
there are
Yes.
limited things in the cytoplasm and those are controlled by the nucleus. Clear? Again, this is mentioned here. This is
Yes.
important. The amount of cytoplasm that can be controlled by the nucleus is limited, so that animal cell may be quite small. That is why animal cell are quite small. And what is the size we have discussed? That is 25 micrometer.
25.
Yes,
25.
exactly.
Yeah.
This is 25 micrometer. Okay.
Micrometers.
Then we have discussed about the we have discussed the cell membrane. Plant and animal cell have common features which relate to maintaining the characteristic of the life. Cell membrane. Cell membrane surround the cytoplasm. You know this is common. Membrane. Whenever there is a membrane, the membrane will cover the content. Okay, the membrane will
Yes.
surround the cytoplasm.
Covered.
It controls the entry and exit of the dissolved substances. Okay, this is very important. The cell membrane. What is the function of the cell membrane? It is semi-permeable. Actually, it is. When we talk about the cell membrane, we say the cell membrane is semi-permeable. permeable. Okay, semi-permeable means it only allow a few substances to enter, and similarly it allow few substances to exit. Okay, it does not allow every substance to enter the cell or every substance to leave the cell. This is not the case.
Yes.
So cell membrane control what will be enter in your cell or what should be exited. Okay, so cell surface membrane surround the cytoplasm. It controls the entry and exit of the dissolved substances and separate the cell content from the surrounding. Okay, these are the two functions
Surrounding
of
of
the cell
contents.
membrane. Yeah, what are the two functions? First is cell membrane, it separate the internal environment of the cell from the external surrounding. Second, it allow only few substances to pass through. Okay, it is semi-permeable in nature. Are you getting this? Just like the gate
Yes.
of our house. What is the function of gate of our house? It separates the inner house from the outer street. Okay, or outer area,
Yeah.
whatever the area is. Second thing is, it only allow when you have security checks on the gate, it only allow those people who have access to that house, right? So similarly, the cell membrane will only allow those substances which have the access to enter the cell. Okay, it is semi-permeable in membrane, semi-permeable in nature. Are you clear
Yeah.
till this?
Yes.
Okay, then we have nucleus. The nucleus contains the genetic material DNA which makes up the genes. What is the function of the nucleus? Nucleus contains what?
The DNA.
Yeah, in the nucleus there is DNA, and DNA, it is made up of genes. Okay, and what are genes? Genes contain your hereditary information. Okay, hereditary information means that information which will pass from one parent to the offspring. Clear?
Yes. Offspring.
From parent to
Offspring.
offspring. Yeah. These carry the coded. Now they have mentioned that the nucleus contain the genetic material DNA which make up genes on the chromosome. This carries the coded instruction. Okay, this carries the coded instruction for controlling the activity and characteristic of the cell. Actually, we will discuss it in detail. That
Yeah.
like the chromosome is like this. One chromosome is like this. Okay, and on this chromosome you have different type of genes. You have different kind of genes on it. Over here, over here. Okay, so in those genes there is information that is encoded in the form of codes. Okay, different codes are there
Yeah.
for specific function. So this is how the nucleus control your cell. Like in the nucleus you have DNA, and that DNA is made up of genes. Genes are made up of chromosomes, and chromosomes have coded information. Okay, these chromosomes have coded information.
Coded information.
Yeah, that information, like what does it mean coded information? That for specific function there will be a specific code, for another function there will be another code, just like the coding
Yeah.
in the computer. Okay, so those coded information, what they do, they control the cellular activity. Okay, they control the
Yes. Cellular
cellular
activity.
activity. Whatever will happen in your cell, it will be controlled by this. Okay, the chromosome
Yeah.
only become visible during the cell division. This is important. We will discuss this in the chapter of the cell division, that how the chromosome appear and how they look like. Till this you just know this about this, that what are the chromosome contain the genetic information. Okay, chromosome contain the genetic information. Okay, and that is present in the they have coded information, and this coded information
Yes.
control the cellular activity of your body. Okay?
Yes.
And
Yes.
sorry, what you are saying?
Yeah, I got it.
Okay. Yeah, one more thing about this is the number of chromosomes in human. You know how many chromosomes are there in human cell?
No.
In one cell. In one human cell. No?
36.
No. Sorry, what you said?
36.
No, not 36. You are close. 46. 46.
Oh yeah, 46. Yeah.
There are 46 chromosomes in the human cell. Okay, there are 46 chromosomes, and these are present in the pair. Okay, you can say 46 chromosomes or 23 pairs of chromosomes. Okay, 23 pairs of chromosome present in the animal, in the human cell, and when they will appear, they will only appear at the time of cell division. They are present,
Yeah.
but you can only see them at the time of cell division. When cell divide, you can just check them at that moment. Okay.
Yes.
Okay, and yes. Okay, then we have to discuss about the plant cell membrane. What are the things which are present in the plant? And the cell membrane is common. Yeah, this inner one. This is cell membrane. This is common in both plant cell and the animal cell.
Yeah.
Okay, then there is
Animal
a
cells.
difference about the vacuole. This whiter portion. This is a large vacuole. Okay, inside plant cell,
Yeah.
plants have a large vacuole. They have
Large
a
vacuole.
large vacuole as compared to the animal, and this is permanent. Okay, this is permanent. This is not temporary, just like the animal. And what is the function of vacuole? It contains water necessary to provide the turgor pressure. Okay, they have mentioned that within the plant cell there is vacuole present, and this vacuole, what is the function of this vacuole? This vacuole control the amount of water within it. Okay, and you know why water is needed? To make it turgid. What is meant by turgid? Turgid means to make it flattened. Okay, to make it flattened. For example, have you ever witnessed that when we water plants, their leaves become erect, but at the night time their leaves bow down, their leaves become flaccid, their leaves become soft. Have you ever noticed turgid leaves?
Yes.
Like okay,
Yeah.
I can show you this turgid. Versus flaccid. Yeah, this is what I am saying. You see this? They have mentioned that vacuole, this vacuole within the cell, vacuole controls the amount of water. It have more water. When there is more water, what will happen? The leaves will be erected like this. You see this? The left side image, high
Yeah.
turgor pressure. Okay, it means there is high turgor pressure. Turgor pressure means there is high pressure, they are erect. Turgor means there is more
Yeah.
turgidity. But on this
Yes.
side, on the right side, you see the low turgor pressure. Why there is low turgor pressure? Because these leaves lose the water. There is less water. Can you see the difference between
Yes.
these two cells? What is the
Yeah.
difference?
Yeah. Like the turgid high turgor pressure makes it like
What is the, you see this cell and this cell, what is the difference in these two cells? What is the prominent difference here?
has like bigger like that high turgor has bigger vacuole and
Yes, it is
the
a
low
large
turgor
vacuole
has low
and it
vacuole.
has shrink
It has a
vacuole.
small vacuole.
Yeah, shrink. Its vacuole is shrink. Its actual size is shrink. Why it is shrink? Because the water is lost from here. Okay, this is known as flaccid state. Clear?
Yes.
Okay. This they have mentioned here, that there is a large permanent vacuole that contain water necessary to provide turgor pressure. Okay, the more water there is, the more turgidity will be there, and the flattened the leaves will be. Clear? Erect the leaves will be.
Yeah.
And what happens when water loses? When there is less water?
It shrinks.
Hmm?
Like the vacuole shrinks.
Yeah, it will, vacuole will shrink, and
There will be
leaves will become
like
flaccid. Okay,
yes.
this is known as flaccid state. Okay,
Yes.
I don't know what you say that, but in Urdu we say that leaves murjha gaye hain. It is flaccidity, and the turgid means
Yeah.
they are erect. Okay.
Yes.
Then one more important thing, what is the difference over here and the animal cell? In animal cell we have discussed that there is glycogen. When you have glucose, the extra glucose which is not used by the cell will be converted into glycogen, but in plants the extra glucose will be converted into starch. Okay.
Starch. Yeah.
The starch is the stored form of glucose in the plant. It can be
Yes.
present in the cytoplasm or the chloroplast. It is the storage
Chloroplast.
form of carbohydrate. Clear? What is the
Yes.
difference between starch and glycogen? Simple. The only difference is that there is structural difference as well. We will discuss that later on. But till this stage, you must know that in animal cell or in humans, the extra glucose is stored in the form of glycogen, right?
Glycogen.
While in the plant cell, the extra glucose is stored in the form of
Starch.
in the form of starch. Okay. And now there is another thing that is known as chloroplast. You know about chloroplast, right? In the animal plant cell. What is the
Yeah.
function of
Yes. Yeah.
chloroplast? We have discussed it.
To make food.
Yeah, it helps to make food. How it helps to make food?
Or
It contains the chlorophyll. Okay. In the
yeah,
chloroplast,
chlorophyll. Yeah.
there is a color-containing pigment that is known as chlorophyll. What is its
Yeah.
function? It helps to absorb light. It helps to absorb sunlight.
Yes.
Okay, we say that plant make their food from the sunlight. How they prepare their food? By using sunlight. They gain energy from the sunlight. Okay, there is a pigment chloroplast that utilizes the chlorophyll. Okay, in it there is chlorophyll. This chlorophyll help to convert the sunlight into chemical energy. Okay,
Yeah. Yes.
and then that energy is used by the plant cell to make the glucose. Clear?
Best way. Yes.